our team to present on the call. We've been working together as a group for at least a year, coming up on a couple of years, doing variant curation for the PTEN expert panel, which was the first expert panel fully approved under the Hereditary Cancer Working Group. And so we hope that we've been a good group of guinea pigs and have learned some helpful tips and tricks that we can pass along to everybody else and share with you, and maybe also get your ideas for how we can improve some of the things that we're doing. So the folks that are going to be presenting on a call, because we work as a team, we're presenting as a team. Myself, Felicia Hernandez, Melody Perpich, and Caitlin Seesock. One time on a call, Jen referred to us as Jesse Mester and the Biocurators, and we thought that sounded like an excellent band name. So I guess once we finish curating all the PTEN variants, we'll be starting our little rock band by that name.
That's great.
All right. So just to take you through what we're going to be talking about, we're going to introduce ourselves. We'll talk about some of the work our expert panel has done to date, touch upon some of the challenges that we've worked through and kind of figured out, and again, hopefully share some advice and tips that'll be helpful for other groups as well. So here are our disclosures. Several of us work for labs, which is how we got into the world of variant curation to begin with. And then we also just wanted to present the full expert panel members. I want to say that it's a fully volunteer-driven effort. Knock on wood, it's been a great group to work with. We've had a lot of fun together and have gotten a lot of good work done. So it's just a pleasure, really, to work on this team. All right, so now we will introduce ourselves. Felicia, go ahead and start us off. Felicia, are you muted?
Okay, sorry.
That's okay. Go for it.
Okay, so I'm a variant assessment scientist for Ambry Genetics, and I'm currently one of the biocurators for the expert panel. I do work on site in Louisiana, California, and currently I have a 6-month-old baby girl. Very busy.
All right, thank you. Next is me. I'm a genetic counselor for GBX's Inherited Cancer Team and coordinate our group, and involved in lots of other ClinGen groups as well. And I work remotely from Cleveland, Ohio, with my kitten Amelia, who is not sitting on my shoulder today, but sometimes likes to help me curate variants. Okay, Melody, you're up next.
Hi, I'm Melody Perpich, and I work in pediatric oncology at the University of Chicago for the past 3 years. I have more than 21 years of experience in clinical cancer genetics. I'm a native New Yorker, but have been living in Chicago since 1987 in the friendly confines of Wrigley Field. And I'm also the hockey mom to 2 teenagers, Izzy and Dylan, whose skills are clearly following a paternally derived autosomal dominant pattern of inheritance with absolutely no distribution from the maternal side of the family.
Okay, Caitlin, your turn.
All right. Hi, everyone. I am Caitlin. Can you guys hear me okay?
Yes.
Okay, good. So I am a clinical variant scientist at Myriad Women's Health, formerly Council. I have been a member of the PTEN VCEP since 2015. I have experience with cancer genetic counseling, and I was also the research coordinator, clinic coordinator for the Cleveland Clinic PTEN study for about 2 years.
Great. Thank you. So we'll be taking turns presenting during this call. But first off, I get to share with you a little bit more about what our group's been up to. Like I said, we were the first variant curation expert panel formed under the Hereditary Cancer Working Group, and have along the way kind of been the test subjects for figuring out a lot of things about how a variant curation expert panel can function, and hopefully we'll keep getting to serve that role as ClinGen keeps we received our formal variant curation expert panel approval almost a year ago in August of 2018 from the SBI. And I get to update this number now because we just had our last expert panel meeting this last week and curated an additional 16 variants. So now we've curated over 100 100 variants to date, and we've been able to resolve classification for several. At the time I put this together, we had 18 conflicting variants that we'd resolved, and 19% of our VUSs we've been able to classify as something other than VUS. We published a manuscript on human mutation in the ClinGen special issue describing our criteria and our pilot curation results, and have presented abstracts at several of the genetics meetings over the last couple of years about our process and results. So one of the first challenges that we came across was figuring out how to do this, how to be an expert panel, how to get the job done. During the rules development process, our expert panel met monthly and we really, kind of hit it hard with meeting on a very regular basis to figuring out how we were going to tweak the ACMG rules, do the pilot testing and everything. But then you get it all done. You publish your paper, and then it's like, well, now what are we going to do? To us, now the real exciting work begins, because now we get to enjoy the fruits of our labor and use all the rules that we worked so hard to develop to classify a whole bunch of variants. So after getting the pilot phase done, we kind of changed tactics and decided to meet on a more relaxed schedule. So now the full expert panel meets on a quarterly basis, still an hour-long call via Zoom. In the beginning, we really didn't know how this was going to go, so we tried to start off slow so we didn't overwhelm ourselves. We were able to get through what I'll call 5 complex variants, and variants that were of uncertain significance by a whole bunch of submitters or conflicting in ClinVar, and 10, quote, easy variants, which we really didn't spend a lot of quality time on discussing on our calls. But over time, we've gotten a lot more efficient. And on our last call earlier this week, we actually kind of flipped the scales and were able to get through 12 of the tough variants, and we didn't have that many easy variants on our docket, but again, we don't spend a lot of time on those. So it's been really great to kind of see our group's efficiency progressing over time. We've just gotten more accustomed to working together, both as a biocuration team as well as the full expert panel group that discusses the hard variants. So some of the things that have helped us along the way and help us, kind of keep the train going. The doodle polls have been, to me as a coordinator, very helpful in being a free tool to get our meetings scheduled. Always trying to think 5 steps ahead of where you are now and working backwards. And then just kind of, rolling with the punches. Things change. The VCI changes. So we just kind of have to, deal with it and enjoy the role that we have with helping ClinGen kind of figure out the process and make it smoother for other groups. So I wanted to share with you the Curation workflow that we've been using that seems to have worked out pretty well. As you can see, the call date, is here, but the work to prepare for the call actually starts about 3 months before, which for our current review cycle is where we happen to be at this very moment. So about 3 months before the next expert panel call, like right after the last one, I, as the coordinator, request the variant prioritization spreadsheet from Stephen Harrison. This is the nice export from ClinVar that gives you, information about if there's conflicting variants, if there's new conflicts in ClinVar, if there are variants that were VUS by multiple submitters, what those are. So this has really helped us prioritize the variants that we want to tackle for review, and also understand which ones are up for re-review. We had our first re-review we had one variant that was VUS after our pilot a couple years ago, and so it was up for re-review. So we rediscussed it this month, and it was great to have a spreadsheet to help us pinpoint, what that variant was, so we didn't have to go searching for it or forget about it. Then, based on this spreadsheet information, I select the variants that we're going to be curating on the next call and distribute them to our biocurators. I sent a doodle poll to the full expert panel members to select the call date and time for 3 months from then, because everybody's schedules are crazy and they will book up. And also poll the biocurator team to select a pre-meeting date for us, which is about 5 to 6 weeks before the call. That's the next one I'll talk about, where we do some more work. And then in the meantime, the biocurators are working on their assigned variants. So then again, about 5 to 6 weeks before the call, we also have some of our biocurators that work for clinical laboratories that share internal data, return some worksheets that we have with internal data that may or may not be utilized for curation. I compile all of this and look at the curation work that the team has done in the VCI, try to put it all together into a nice slide set for us to discuss each variant. And then we meet to discuss the criteria and the classification that we've applied for each variant and decide which ones require really detailed full expert panel discussion. If there are specific papers that we really need the expert panel members to review, we have some different subgroups. We have a functional subgroup, people that have a lot of expertise with functional assays. We've got a clinical subgroup and a computational subgroup. So most often it's the functional subgroup that gets hit. We might find some papers with conflicting or evidence we're not sure about how to interpret that we want them to especially focus on and review and maybe respond before the call or have a heads up about for a discussion on the call. And I know Caitlin will be talking about this more, but we have been utilizing a dual curation system to date. We are going to be trying out a single curator system soon. So after we review and get all this preliminary work done, the expert panel members requested that they get the slide set that gives some information on the variants we're going to be discussing at least 4 weeks before the call. So we put that together and send out so that way when they have the time, they're able to review it and think about it and see if there's any points of discussion that they want to bring up on our call. During the call itself, we, as a Biocurator team, take turns presenting each of the variants that require full expert panel discussion. And then after the call, myself as the coordinator, I finalize the interpretations in the VCI, alert Stephen that they're ready for export. We're helping Stephen and the VCI folks pilot the process of getting variants directly from the VCI into the ClinVar spreadsheet format. And then I review the export and submit the submission to ClinVar. Okay, so Caitlin, go ahead and tell us about how we figured out dual curation.
Okay. So currently, as Jesse mentioned, we currently curate with a dual curation process in the VCI under a single variant affiliation of the PTEN VCEP. So in the beginning, we actually did not use this method. In the beginning, what we really tried to do is we tried to basically figure out how to use the VCI. We put variants into the VCI and used like the test version of the VCI. I'm sure everybody has used the test version and kind of taken a look at that. But what we really did in the beginning was we used our own name, so no affiliation within the VCI, to curate our variants in the beginning. So we did that, one, in order to help us learn how to use the VCI and learn different ways of how we want to put information into the VCI, how we want to record things. But we also wanted to do curations under our name, our own affiliation, really also to help kind of blind our classifications and see how we were curating variants and kind of how each curator's classifications aligned or did not align with each other. So today, like I mentioned, currently what we do is we curate all under one affiliation, so we all curate under the PTEN VCEP affiliation, and we do use the dual curation process. So basically what we do is we curate under the PTEN affiliation, we each make comments, so each variant is assigned to 2 different curators, and we basically both input information into the VCI for those variants. If there are comments that are already in the VCI, then the biocurator who is inputting information into the VCI can agree with what the other curator has already written or put into the VCI. They can disagree. They can add information. What we will do is we'll put in our initials with any of the added information, so that way when we go back into the VCI and we're looking at that information, we can know which biocurator was the one that kind of added that comment or may have changed information. And then with that process at the end, what happens is Jesse, as our coordinator, once we have our meeting and sign off and review all of the variants with the whole entire team, Jesse will go ahead and review all of the information that is in the VCI and kind of do anything that she needs to do to get that ready for submission to ClinVar.
Alicia, I'm going to go over one of our next challenges we face was getting used to gene-specific criteria. So when you begin as a curator, if variant curation is currently your job, you kind of have to leave behind the guidelines used by your current workplace and then adopt those established by the expert panel. I think this is relatively seamless in my case, since many of the guidelines were similar. But I just wanted to go over a few differences I encountered. And how I was able to remedy those just to avoid any discrepancies in classifications in the future. So in order to allow for consistency, the PTEN VCEP established a functional experts subgroup, and this subgroup allowed for the assignment of PS3 and PS3 supporting, as well as benign and benign supporting criteria for functional studies. And this allowed for consistency, which we currently use. And then the subgroup comes to a consensus regarding these criteria, and just some of the things that they discussed when they were establishing whether to assign criteria or not was they looked at the paper and we set thresholds for the level of reduction in activity that needed to be seen, for example, enzymatic activity, the number of biological replicates that would be needed, proper controls, if there was a decrease in protein expression, what level that would need to be, whether cellular localization was affected, and they also discussed other aberrant phenotypes in comparison to wild type, and any impact on splicing. So these were papers that come up very often in our curation, and they were vetted by this subgroup. And I believe it's really great because it allows for consistency when we're doing our curation. Okay, so another difference when curating variants was in silico models. So since there was a lack of established likely benign and benign variants that were missense in PTEN, it was hard to test the accuracy of in silico predictors in these cases. But I feel in the future this will change as we start to curate more variants. Another difference was that for PM1 specifically, which is located mutational hotspot or critical well-established functional domain, the PTEN expert panel assigns evidence to specific residues within catalytic motifs. Ambry is similar in that way as well. It's just that they would need to first be assigned to a structural biologist for evaluation before weight can be given. For proband specificity scores, or PS4, so a scoring system was developed by the expert panel known as a Cleveland Clinic score, and this currently wasn't, when I started, it wasn't being used in Ambry, but we're slowly implementing this as a way to count probands for weighing evidence. Another discrepancy was the allele frequency. So the PTEN expert panel set an allele frequency for BS1, and we were able to adopt this at our lab as well. And then for a missense variant in a gene that has a low rate of benign missense variant. So PTEN is a highly constrained protein according to NOMAD or EXACT. And so the expert panel does implement PP2. And this was a discrepancy with our laboratory, but expert panel was justified in this. In using this rule just because at the time the rules were established, no missense variants had been classified as benign or likely benign. Okay. That was all I wanted to go over.
So although Jesse discussed curation workflow, I'm going to focus a little bit more on specifics of making time to do biocuration in our busy work lives and work weeks. So of course there's a learning curve. So the time to curate is really going to depend on your current knowledge, specifically of gene-specific criteria for variant curation as it relates to your gene, your level of expertise. Maybe if you work in a lab already and you're doing this as part of your regular work week. This is going to be easier for you. The level of training and experience with databases, for example. So we have 3 members of our team that do work as biocurators. I'm the only member currently that has only clinical cancer genetics experience. So that impacted my personal learning curve, but it's been a steady improvement, I'd like to say. Familiarity with available search tools and relevant go-to publications are important, and understanding of the functional studies that are available. The understanding of principles of genetics and specific clinical diagnostic criteria. And of course, the more you practice and the more experience you have, the faster it will be to do biocuration of the variants. So like a roller coaster, I kind of acknowledge it to be like. We kind of work in fits and spurts. There's sort of a slow climb to the peak, which I view as the heavy workflow time. And then we coast down a bit when we're getting ready to present to the entire group. And then we get a chance to level out for a period of time. And throughout the cycle, there are periods of breaks, so it's not constant work. So their time between periods of biocuration, the time between the periods is about 12 weeks, as Jesse previously mentioned. And once our biocurators have met online and finalized the work and the slides, we have about 5 to 6 weeks in a sense that we're off before we actually present to the entire expert panel. And then once the presentation has occurred online, we have about a 2-week vacation period until the next group of variants is distributed to the group. So this slide sort of just summarizes what I just spoke about, but again, there's a 12-week cycle with breaks. Once the expert panel call happens, we have about a 2-week break until the variant list is sent out, the new variants are distributed, and then we have sort of the greatest period of workflow in that 4 weeks after the variants are received. And we do our biocuration then and meet online to review the curation work that we've individually done. And once that's completed, there's about a 5 to 6 week break, again, before the expert panel call happens, at which time we might add additional internal lab data that we're able to receive. And Jesse puts together the slides for us. So the specifics of making time per variant is something to think about ahead of time, depending on how many variants we have in the distribution list. It can take between 30 minutes per variant to up to 2 to 3 hours, depending on, again, the individual level of experience and the amount of information that's available on the variant to look at and to review. So that includes a significant review of the literature and the internal lab data that we're able to acquire, applying all the criteria, and then uploading it to the VCI. So in combination, now that we're doing about 6 variants per cycle, it can take about 3 hours to as much as 12 to 18 hours done, again, over that 12-week period, but focused mostly on that 4 weeks of intense biocuration time. So it's important to keep your meeting dates in mind, your personal obligations and work commitments, and schedule the time to curate. So depending on how you work, it might be better to spread it out over that 4 weeks, or to, if you know you have more time in a particular week, you might focus your more time during that specific week. Our team coordinator puts the slides together for the PTEN expert panel presentation, and kudos to Jessie for doing all the great work that she's been doing for us.
The one lady went through the different BPs and stuff, but that's it. That's it?
Yeah.
That's it? Well, it might come back to someone.
If you work part-time like I do, you can schedule time on days off to work on biocuration, but evenings and weekend time might be necessary for biocurators who work full-time that can't fit it into their work week. But hopefully you enjoy the time you spend biocurating, either honing your skills or learning new skills involved in biocuration, and interacting with your team, or in our case, our band, which we do enjoy. So we always do need more curation help. The benefits of having additional team members is that we can reduce individual workload. or ideally increase the total number of variants that can be biocurated and reduce the time that it takes to reclassify the variants. The more lab people specifically that we have will allow us to have more internal data available and result in easier reclassification and also reduce the time to that reclassification. Another idea is to have tier 1 curation. This sounds fairly helpful if we can identify curators in the community with varying levels of genetic expertise that can be assigned roles of like specific targeted projects like documenting information from large papers and assisting the expert panel. So how can we recruit biocurators? I wanted to acknowledge that ClinGen is working very hard to recruit biocurators through various meetings and on their website and conducting volunteer sessions at ACMG and other meetings. In my experience, I kind of view myself as an accidental curator in a sense because I met Jesse Mester at the ASHAG meeting about 2 years ago in front of her poster, and I asked her questions about how she went from clinical genetic counseling to working at GeneDx and what it took to be a biocurator. And she informed me that, you volunteer and you can learn about, the techniques involved for biocurating. And she adopted me as her trainee, and that's how I became involved in our group. So it's important to share your experience and discuss the various biocuration opportunities that are out there and approach colleagues when you can. There's also the possibility of training genetic counselors or PhD students and lab colleagues to do it, and then consideration of advertising through the graduate programs, the various listservs, at meetings, special interest groups, and of course by word of mouth. And I just want to mention there was actually a play called The Accidental Curator at the Steppenwolf Theater, but I did not see it, but I did see the advertisement for it, so I thought that was a great slide to add.
All right, what else has helped us keep going? So I think everyone will agree that we have a great team, a great curation team, a great PTEN VCEP group. So it's definitely a great balance of both curation and fun. We know that our entire group and, pretty much everyone who is participating in these groups are volunteering their time. And we recognize that this is a great way to meet new people, make new professional connections, make new friends. It's also a great way for all of us to, have some professional development, have learning opportunities, learn things that we don't necessarily know. If you may be from a strictly clinical background, it's a way to maybe learn more about molecular and variant assessment information, or if you're from a lab background, this may give you an opportunity to develop more clinical expertise. Also things like publication and abstract credit, meetings, meeting others, trying to get more volunteers, and having outreach at meetings is helpful as well. For any of the genetic counselors on the call, PACs are available until December of this year for volunteering service to a genetics-related organization. And ultimately, it really feels good to contribute to patient care, and helping out really feels like a great way to give back to the genetics profession in the community participating in this group.
Sorry. Okay. So finally, we have a lot of folks that we want to thank. Certainly the expert panel members, I think, place a lot of trust in us as the biocurators to do all this groundwork that goes into doing the curation for the variants that we're working on. So thank you to all of them, especially Karis and Naduri, our co-chairs, for trusting us and letting us take the reins to figure this process out. Also want to acknowledge Tina Pesserin, who's I call a backup biocurator in case we're forever short during a cycle. And also Raj Ghosh, who helped out with curation during the early pilot phase of our group. And finally, we also want to thank lots of folks on the ClinGen staff, especially Laura, Stephen, Deb, and G, for all the help that they do to make sure that our group can get our work done. So with that, we wanted to open it up to any questions that anybody has. We're all happy to field them as a team.
Great. Thank you all so much. I think it's just really nice to hear from a group who is so far ahead and looking at how you're organizing everything. I think that's just really helpful. So I'm going to unmute all of the PTEN presenters. And I think there were a couple of questions from Dimitri Sonkin and also from Kristen Maloney. Is that right? You guys should be unmuted now if you want to ask a question. Right, or if anybody does have a question for the group, you can put it in the chat box.
I can't see the chat box, Jenny.
So it should be, if you click on the screen, there's a little bubble.
Oh, there we go. Here we go. Chat. Yay.
Dimitri and Kristen, did you have a question?
So this is Dmitry. So I have a couple of questions. So one question is, at what point does PTEN submit variants to ClinGen evidence repository?
So my understanding is that because we are curating in the VCI, that happens when Stephen does the export. I personally don't have to do any work to make that happen, but I don't know if Matt or somebody else that's affiliated with the VCI is on the call, but my understanding is that because we curate in the VCI, then that information populates in eRepo. But am I wrong?
Let me unmute Matt. I think Matt is on the call.
Can you hear me?
Okay, yes, I can hear you now. Yep.
Can you hear me? Oh, good. Okay, I'll say that again then. I was going to say that, yeah, at the moment you submit the files to Stephen as part of your ClinVar submission process, and he submits them to Ronak and then at Baylor, and then they get showed on the eRepo. But in future, it will be part of the ClinVar submission process. That automatically, when you submit to ClinVar, it will automatically at the same time be submitted to the eRepo.
Yeah. So, okay. So what I'm hearing is that right now it's no extra work for me as the coordinator, and in the future it still won't be any extra work for me as the coordinator. So that's great.
Right.
Okay, so the second question is, you mentioned in the beginning that currently there are 98 variants which have been interpreted by PTEN expert panel. Currently in the evidence repository and ClinVar there are 74, 75 variants. Is it possible to get complete list with all 98 variants interpretation from the PTEN expert panel? Yeah, so the reason that not all of our variants are in ClinVar, it's not our fault. It's because Stephen was waiting until some updates were going to happen with the export process to do our next batches. So thank you for reminding me. I need to bother him once he is back from vacation and see where we are.
I can answer that as well if you can still hear me. Yeah, so Stephen was relying on us releasing like a button within the interface that can be used to do the export, and that will be in place this weekend. So, and Stephen's away this week as well. So a good time to harass him will probably be Monday, because then the functionality will be in place.
Perfect. Right when he's back from vacation. Yeah. So Dimitri, just so you know, we're trying very hard to stay up to date and make sure that everything in ClinVar is current. But as the guinea pig group that's helping pilot this process, we're also happy to be patient and wait a little bit to have things submitted to ClinVar, because it's going to be helpful for us figuring out how to use these tools to their